you want to understand how Parkinson's disease works, it's important that you understand how the nervous system works. So essentially the nervous system includes sensory receptors that receive information, interpret it, and result in some sort of motor output. The major components are the central nervous system, which includes the brain and the spinal cord. And this is responsible for integrating, processing, and coordinating sensory data and major commands. And then from there, there's the peripheral nervous system, which includes all the neural tissue outside of the CNS, and that brings information to the CNS and uses motor division to carry out motor commands. And then from there, the nervous system branches out even further, and there's the somatic nervous system, which is responsible for controlling skeletal muscle contractions. And it's interesting because Parkinson's disease initially affects the brain, but the effects of it manifest more in the somatic nervous system with the lack of muscle control. So, I'm pretty sure we all know what the brain does, but in case you don't, it controls all functions of the human body, it receives and interprets information from the outside world, and it controls many responses to stimuli. The parts of the brain that we just like to focus on mostly for this presentation is the basal ganglia, which is right over here. It's basically a collection of structures within the brain, and it's associated with many functions, including the control of voluntary movement and eye movement. And it's interesting because those are the things that are generally the most affected by Parkinson's disease. And then now that we've kind of had more of a zoomed out picture on the nervous system, we have to zoom back in a little bit, and this is the most basic component of the nervous system, which is a neuron. So on the incoming end of the neuron, there are the dendrites that receive signals from other neurons, and then there's the soma, or the cell body, and the nucleus inside of it. And then there's the axon, which is comprised of Schwann cells and the myelin sheath. And then at the end of the axon, there are the axon terminals, or terminal buttons. So there's like different names for it. And then that's where the neurotransmitters are released, and then they cross the synapse, and then the signal continues into the other end of another dendrite, and then just keeps going through the body. The signals pass and like jump through the nodes of Ranvier, which are like the depolarized regions between the Schwann cells, and the myelin sheath insulates, protects, and speeds up the process of neurotransmission, but I'm going to stop talking about those because myelin sheaths really get on my nerves.
Okay, so this is basically just an overview of how neurotransmission works. You guys probably know all of this anyway. Like Avni said, the electrical impulses will jump from one node to the next in the depolarized regions. So obviously your resting potential is at negative 70 millivolts because you have like they're both positive ions, but you have fewer positive ions on the inside of your neuron than outside, so that's why we delineated it with negative voltage. In this diagram, it's just that the orange, well the orange is going to be, the green is going to be the sodium potassium pumps, and then you have sodium on the outside, so the yellow is going to delineate all the sodium ions and then also the sodium channel, so passive transport, and then the orange is going to be the potassium ions and then the potassium channel. So what happens is you'll have like ligand-gated channels, and when a ligand attaches to your channel, you'll have like the sodium flood in and then reach your your threshold potential, and then this electrical impulse will just propagate down the neuron. You guys know this, and then the previous channels will close. Once you hit positive 35-ish, the potassium channels will open, and then you'll do hyperpolarization, and then active transport and repolarizing, reinstating the correct negative 70 millivolt charge in the neuron. So basically we talked about electrical impulses, but the end goal of the neurotransmission is for you to generate a chemical signal at the end, where Avni said before, like the terminal buttons. So this is actually the neuromuscular junction, and it's the point of signal like connection between the neuron and then the postsynaptic membrane, in this case a muscle, which is like specific to our disease. What happens here is in the presynaptic membrane, you'll have like calcium channels, and when they open, your terminal buttons have like neurotransmitters that will be released by exocytosis and then received on the postsynaptic membrane. So here, like neuromuscular junction, here it connects to your muscle fibers, and it will trigger like muscle contractions as your calcium ions are released. This is an overview of muscle contraction. Do you want me to keep going?
Maybe just finish your part and then
Yeah.
eventually get.
Okay, so muscles, the contractile units are sarcomeres, and then you have thick myosin filaments and thin actin filaments, right? So like I said before, when calcium or neurotransmitter is released from the neuron and at the neuromuscular junction, calcium will be released, and then basically, can I just finish this on Monday? Okay.